Hello achievers, welcome to Team MDS Achievers, myself Dr. Vikram. Today we are here for the discussion on the topic of glycolysis. Firstly you should know that every multicellular organism has a cell, which is considered as a structural and functional unit of a multicellular living organism, right? So thereby all the biochemical reactions that are occurring in the cell are to be classified into two types, that is anabolic or catabolic. The catabolic is the one where the energy is being generated from it, that is glycolysis and Krebs cycle are going to be catabolic pathways where the energy is being generated from the molecules. So firstly you should know the significance of the glycolysis. That is like, see, all the movements in our body, that is speaking, masticating, or all the our skeletal tissues come in contact with the muscle tissues, and thereby movement is brought. And when you go the structural and functional unit of the muscle, that is sarcomere, where the contraction of the muscle occurs by the attachment of the actin and myosin together and causing the movement of the muscle or contraction of the muscles. This is facilitated by the ATP, that is the energy currency of the body or energy currency of the cell. जलवा है हमारा. So to generate this ATP, the glucose should undergo the biochemical reactions to generate the ATPs. Through the either glycolysis, which generates only 4 ATPs, and net gain is going to be 2 ATPs. Whereas when you go to the mitochondria, that is Krebs cycle, and also the ETS, that is electron transport system, we are going to generate about 38 ATPs, which is very high, right? And net gain is going to be 36 ATPs. So in order to generate such a high amount of energy, we should know the molecule should go through the glycolysis, link reaction, Krebs cycle, and ETS. So glycolysis being the first step in the catabolic reactions to generate the energy. So it is a very good and clinical significance is present. So to understand the concept also. Now I will explain in the story manner. That is like, firstly we are eating the food. Okay, we are eating the food enters into stomach, and the food will be getting digested. That is like starch is going to be digested into the carbohydrates. That is like glucose, fructose are going to be formed, right? And results are galactose or the different. That is, these are the monosaccharides. See, these monosaccharides are being absorbed into circulation, and they are transported to the cells, where this glucose will enter into the cell. That is like, they says, now I am being digested and I came into the body, so where should I go and act my activities present? So once the glucose enters into the house, that is like, it goes and knocks the door of this cell. अबे मेरे को तो अंदर लो. It actively transports the cell in glucose into the cytoplasm of the cell. So what happens once it enters into the cytoplasm of the cell? It is catabolized, or it undergoes a reaction with the exokinase enzyme, that is activated by the magnesium plus 2 ion, and the enzyme code of this exokinase is 2712. What happens is, glucose enters and it needs the exokinase enzyme. What says, okay, now you have to go a reaction called as phosphorylation. I will add one of the atom to you. So it says, the glucose says, okay, I'm good with you. So add that is phosphate group to me. So what happens? This exokinase with the glucose will add an exokinase. This exokinase will add the phosphate group to the glucose and result in the formation of glucose 6-phosphate, and this reaction is called as phosphorylation reaction because the phosphate group is being added. Okay, and here the ATP is consumed because the phosphate group is taken from the ATP. ATP is made up of adenine diphosphate with inorganic phosphate group. Both together is called as adenine triphosphate, and which is the energy currency of the cell, right? So thereby the phosphate group is added to it and results in the formation of glucose 6-phosphate. Once the glucose 6-phosphate is formed, it undergoes an isomerization reaction, resulting in the formation of the fructose 6-phosphate. Fructose 6-phosphate will undergo once again phosphorylation. That is like, see, the fructose 6-phosphate once again phosphorylizes and results in the formation of fructose 1,6-bisphosphate. Because it has in two inorganic phosphate groups, it is called as bisphosphate. That is, it has in two phosphate groups. Okay, and this reaction is a very important reaction, and this is a rate limiting step because this is irreversible reaction and it is catabolized by the enzyme called as phosphofructokinase. This is one of the very important enzyme which is called as rate limiting enzyme, because once this step crosses, there is no way going back. It is only forward. And phosphofructose kinase says that
ఇదే నా మాటే శాసనం.
that is fructose 1,6 bisphosphate will undergo the cleavage reaction. See, fructose is in a 6 carbon molecule. So it undergoes splits or cleavage reaction in the presence of the aldolase enzyme and results in the formation of two molecules of 3 carbon compound. As it is 6 carbon, it is converted into 3 carbon molecule of 2 molecules. Okay, so that is G3P, which is 3 carbon compound, and DHAP is 3 carbon compound, which are formed from 6 carbon molecule, that is fructose 1,6 bisphosphate. Simple enough, right? See, DHAP is not at all useful, so that is why it is isomerized to form the G3P, that is glyceraldehyde 3-phosphate. So these two molecules of G3P will further enter into the reaction called as oxygenation reaction. See, oxidation is of 3 types. That is, first thing is the addition of oxygen to the molecule is called as oxygenation, first type of oxygenation. Second is the addition of removal of electrons from the molecule is called as a second type of oxidation reaction. And third one is the removal of hydrogen atoms. Any of the 3 steps is occurring, it is called as oxidation type of reaction. And same wise in the vice versa, we can say is reduction reaction also. Okay, so the step that is occurring over here is the removal of hydrogen atoms from the G3P, so thereby resulting in the formation of the 1,3-bisphosphoglyceric acid. Okay, see, 1,3-bisphosphoglyceric acid has 2 ATPs inside it. Okay, two phosphate groups inside it. So what happens? It undergoes the dephosphorylation. That is, the ATP is released or ATP is formed by releasing the inorganic phosphate group from this 1,3-bisphosphoglyceric acid. Okay, simple. This is where you people have not to be confusing about it, which is a 1,3-bisphosphoglyceric acid. The dephosphorylation reaction, that means the phosphate group is being removed. The reaction name itself is self-explanatory. So the phosphate group is released and it is taken up by the ADP, that is adenine diphosphate, and results in the formation of adenine triphosphate. See, ATP is the energy currency of the cell, which is made up of ADP and IP. Okay, so this is the basic structure of the ADP. So the as the ATP is donated to this ADP, so thereby it is results in the formation of the 3-phosphoglyceric acid. It has a 1,3-bisphosphoglyceric acid, and as one group is being removed, it results in the formation of 3-phosphoglyceric acid. Simple enough. Then this 3-phosphoglyceric acid undergoes mutation reaction, that is in the presence of mutases enzyme. That is, see, it is a 3 carbon molecule, and the phosphate group that is present in the third carbon atom will be transported or translocated to the second carbon atom, and that is why it results in the formation of the 2-phosphoglyceric acid. Okay, simple enough. That is mutases enzyme, which is changing the phosphate group position from the third carbon atom to the second carbon atom. Okay, simple. Then it undergoes the reaction called as in the presence of enolase, it undergoes a reaction called as dehydrogenation. That is, the water molecule is removed from it and thereby results in the formation of phosphoenol pyruvate. Phosphoenol pyruvate will further undergo dephosphorylation. That is, the phosphate group is removed from it and results in the formation of ATP, and results in the main molecule that is formed is pyruvic acid, and the ATP are the two things that are formed after this reaction. So the ATPs are formed in the 2 steps, that is in the 7th reaction and in the 10th reaction, that is the final reaction. Okay, and the ATP that produced are 4 ATPs are produced, because in the 7th step 2 ATPs are produced, and in the 10th step 2 ATPs are produced, and 2 ATPs are consumed in the first and third reaction, where glucose is converted to glucose 6-phosphate in the first reaction, and fructose 6-phosphate is converted to fructose 1,6 bisphosphate in the third reaction, right? So thereby these are the steps where the fructose that is phosphate groups from the ATP is going to be consumed. So totally the net gain of ATP is 2, because 4 are produced, 2 are used, so net gain is going to be 2 ATPs. Sorry. Okay, simple enough. There is nothing you need to worry about it. 4 ATPs are produced, and from it the 2 ATPs are already used in the first and third reaction, so that's why it is called net gain is going to be 2 ATPs. Simple enough, and there is no need to worry, right? Simple. The glycolysis is completed. But suppose like a person is running from a 100-meter or 200-meter mile, suppose. Let us assume that the person is running for the 200-meter mile and he has a stamina about for 100 meters only. So the person running from the 0 meters to the 100 meters will be consuming all the energies under the aerobic pathway. That is, the glucose is being consumed, glycolysis, then link reaction, then Krebs cycle and ETS, and totally 36 ATPs of net gain. I am speaking about net gain ATPs. Total ATPs are 38 ATPs, but net gain it is going to be 36 ATPs are produced. But what happens after the 100 meter miles? After 100 meters, its stamina is going to be consumed, or its energy is going to be depleted. So it enters into an oxygen is being supplied to the cell is decreased. So what happens? It undergoes anaerobic pathway. That is under the absence of oxygen. The glycolysis is the only step where it occurs in the both aerobic and anaerobic. But under anaerobic condition, here only, that is like after formation of pyruvic acid, the step will stop and results in formation of the lactic acid from a pyruvic acid. This is what happens. Suppose a person is crossing the 100 meters mile, his stamina is get depleted and the oxygen supply to the cell has been depleted, so it undergoes the anaerobic pathway and results in the formation of the lactic acid. And this happens, and thereby muscle fatigue will occur. Simple. This is the way you have to remember this glycolysis. What is the significance is because like all our muscles have this pathway that is going to produce energy, because see, actin, myosin are the binding sites or the contractile units of the muscle, right, that are occurring in the sarcomere. So this now to binding of the actin and myosin, the ATP is required, which is required for the contraction and muscle movement in coordination with the skeletal tissues. This is the why we have to study the glycolysis. And also in the bacteria under anaerobic condition, the byproduct, the end product that is going to form is ethyl alcohol, and certain bacteria produce another group of molecules also. The most of the commonly the ethyl alcohol is being produced as an end product in the glycolysis. So the significance of this studying is because under aerobic condition how the cell behaves, and under the anaerobic condition how the cell behaves. We should be know, because see, bacteria are the main agents which are causing all the tooth problems. That is like if it is present on the occlusal surface, it is going to cause cavities. If it is present on the, that is like in the marginal gingiva or gingival tissues, it is going to cause a periodontal problems, right? See, this is why we have to study the mechanism of the glycolysis. This is simple enough, and we have discussed in story manner, that is like so that you can understand the concept very easily. And now you might have a doubt like why we have to choose the MDS Achievers Academy. That is like simply we are bridging the both theory knowledge with the clinical knowledge, and that is going to be give you a success in your life. That is what. Thank you. This is Dr. Vikram signing off.